We are at 3:31, so let's get started. We are thrilled to be here to represent the DREAM team, the Drug Repurposing for Effective Alzheimer's Medication study. And it's a large multidisciplinary study involving 3 centers and the National Institute of Health. And so I would, at the outset, like to thank all our collaborators who will be named in some slides. But I would also like to introduce the speaker panel today. So Dr. Madhav Thambisetty is really the person who brought us all together. So he's the chief of the clinical and translational neuroscience section laboratory and behavioral neuroscience at the National Institute on Aging. And he has been interested in doing drug repurposing work for Alzheimer's for a long time. By training, he's a pharmacologist and MD neurologist, and so he has done some basic pharmacology work, and then we collaborated together to bring in pharmacoepi concepts. Unfortunately, he had some last-minute travel issues, so he's not able to join us, but we will have Dr. Siegel as a replacement. So Dr. Siegel is a professor at Johns Hopkins University, and she was a key collaborator, and she's a well-known pharmacoepidemiologist and internal medicine doctor. So she will be taking us through some of the hypothesis generation steps that we followed for the DREAM study. And then Dr. Toby Gerhard is a pharmacoepidemiologist, professor at Rutgers, and also the president of ICPE. So he will be talking about some of the pharmacoepi challenges that we faced and tried to solve when using Alzheimer's as the outcome, because as many of you might recognize, it's not an easy outcome to study in pharmacoepi studies. And finally, my name is Rishi Desai. I am last and least assistant professor of medicine at Harvard Medical School, and I will be going over 2 key case studies ranging from a success story and then a story where we really did not see an association, but still very, very instructive. And so I will be talking about the two results that we have published from DREAM studies towards the end. And so with that, I welcome you all, and I will hand it over to Dr. Siegel.
Thank you. So glad to be here to present on behalf really of Dr. Thambisetty. So I'm going to start by talking about really the motivation for bringing together this DREAM team. Dr. Desai introduced the team, and he minimized his role in this project, which was really, really the design and the conduct of many of the analyses. So Dr. Thambisetty, as stated, is a neurologist and a clinical pharmacologist, and has built his career about understanding the mechanisms of pathways to Alzheimer's disease with the goal of drug repurposing. So he reached out to me about the feasibility of doing drug repurposing studies in big data and whether that's feasible. So I'm going to present his slides today, and these are a subset of the slides that he presented to his clinical colleagues at the National Institute on Aging to really get them on board as well. So his goal is to establish a pipeline for identifying drug targets in Alzheimer's disease through systems biology, which is his expertise. So there's really 2 pathways that he's been most interested in. One is metabolomics for bile acid signaling pathways as a drug target in dementia, and the second is about cytokine signaling as a targetable pathway. So in selling this to his colleagues, he made the point that a lot of trials for Alzheimer's disease drugs have failed over the past decade. And in this review article, they talk about failures in disease-modifying treatments as well as failures in symptomatic treatments. He notes that a lot of the advances in technology have just not translated into treatments. The ApoE4 allele was first identified in 1993 as being an Alzheimer's disease risk, but really the last symptomatic Alzheimer's disease treatment until the recent biologics was in 2003 with memantidine. There have been additional GWAS studies, genome-wide association studies, that have identified a lot of new intriguing loci, but nothing that's resulted in treatment yet. So when he talks about big data, it's really proteomics and metabolomics and transcriptomics, right? Not quite our kind of big data. But the promise of these techniques really haven't yet yielded targets. So he believes that this is the process for identifying drug targets. Start with well-characterized aging cohorts and do deep phenotyping of their physiology to identify biologically relevant drug targets. Then use converging evidence from different cohorts using different omics techniques to test the biologic plausibility of these drug targets, and then do very context-specific clinical studies to help interpret these data to identify plausible drug targets. So he illustrates with this slide. Begin with the unbiased omics studies, deeply characterize the cohorts as part of discovery, validate across multiple different cohorts, and then we come in, right? Pharmacoepidemiology to test whether there's a signal that these targets might be relevant for drug targeting. Then, if so, it can proceed to preclinical studies and clinical studies. So the early work in the deep phenotyping was done in the Baltimore Longitudinal Study of Aging, which is a cohort established in 1958, more than 3,000 deeply phenotyped participants. Currently, there's more than 1,500 participants. They've been undergoing neuroimaging since 1994 with brain MRI, with PET scans, with PET amyloid scans, and more than 10,000 cognitive tests. So based on work starting with Baltimore Longitudinal Study on Aging, he's identified bile acid signaling as a potential target. There's 3 major routes of bile acid synthesis, and early work has shown that low primary bile acid synthesis in males is associated with higher brain amyloid burden, faster brain atrophy, and faster accumulation of white matter lesions. There's also supportive information from gene expression where bile acid receptors are altered in Alzheimer's disease in a sex-specific manner. Bile acid sequestrants are, of course, lipid-lowering agents, were the primary agent before statins, still are used often in conjunction with statins. And one of the early studies that his colleagues did was to test whether exposure to bile acid sequestrants alters the risk of dementia. The first study was done in CPRD data, and they used a cohort design and found that bile acid sequestrant use was associated with marginally increased risk of vascular dementia, so not Alzheimer's disease, but vascular dementia, and there was a dose-response relationship. So he concludes from these studies that bile acid receptor signaling is a plausible drug target in dementia. Now, similarly, he believes that cytokine signaling is a potential drug target. The ApoE4 allele is, of course, the most robust risk factor for Alzheimer's disease. And the JAK-STAT pathway, which is related in some way to the ApoE4 allele, is a potential drug target. So this, too, he believes is appropriate for drug repurposing. This has also been demonstrated in gene profile expressions, where the cytokine transducer STAT3 is associated with evolution of Alzheimer's disease pathology and symptoms. And happily, there are drugs already that target the JAK-STAT pathway. disease-modifying drugs like tocilizumab, which is an IL-6 blocker, TNF inhibitors, and tofacitinib, which is a JAK1/3 inhibitor. So, based on his phenotypic work, he came to us and said, let's do what has now been called the DREAM study. So this is a 2-year, multi-center study for identifying novel repurposing candidates for Alzheimer's disease using Dr. Thembasetti's deep molecular phenotyping, and then bringing it to us for big data analyses of real-world clinical outcomes. So he literally came to us with a list of drugs that he suggests might be appropriate for drug repurposing, and suggested that we study his big list of drugs. So this brought together researchers at Harvard, Johns Hopkins, and Rutgers, and 2 real-world clinical data sets. So Medicare data, which Dr. Desai led the analyses of, and CPRD, which Dr. Gerhard led the analyses of. I came along for the clinical input as a practicing internist. I was helpful in picking the comparators for these drugs of Dr. Thembasetti's. So the hope is that the converging results from these independent analyses will ensure reliability of the findings and support their confirmation later in trials of these perhaps appropriate retargeted drugs. This is Dr. Thembasetti's slide that was published in our first published paper, really laying out the different pathways that he thinks are targetable and may really support drug repurposing. So, how did it go? Bring up Dr. Gerhard.
Thank you, Jody, for setting me up so nicely. So I will be talking about, now that we had this list of drugs and basically this outline of what we were supposed to do, how did we go about it, how did we think about it, and obviously this was an effort that involved really all of us. It involved Dr. Schneeweiss, Dr. Siegel, and just talking through the different challenges and debating the designs, and this is basically a very brief summary of kind of what we arrived at and a little bit of our thinking. As disclosures, I have funding as PI and co-I from NIH. So I think to start this, I think it's important to realize that drug repurposing studies aren't some magic new type of study. It's just a pharmacoepi study of drug benefits. So all the standard considerations for pharmacoepidemiological studies that we know apply very much here. The issue with drug repurposing studies is a bit that they're often executed by non-epidemiologists. So it's oftentimes basic scientists that then maybe reach out to a data scientist or know somebody that has a database, and then they try to do it basically on their own, and it's really a big credit to Dr. Thembasetti that he really reached out to various pharmacoepidemiologists to put this project together. Because what you often see in these studies is really things that you don't want to see in pharmacoepi studies: non-user comparisons, never ever comparisons, prevalent user studies, all those things that really bring a bad reputation to the whole idea of these drug repurposing studies and real-world data. What you need is really, and I believe what we need is what we have tried to assemble here, multidisciplinary collaborations with basic scientists. The work of Dr. Thembasetti is certainly out of the wheelhouse of probably all of us here. Clinicians, because I think while pharmacoepidemiologists have often areas, if they're not themselves clinicians, areas of interest and specialty, when doing drug repurposing studies like Alzheimer's disease and studying a battery of different drugs, by definition, this involves treatments of a large number of diseases. So having the clinical input for each of these diseases to think through comparator choices and confounder choices is really critically important. And so is pre-registration, just again, particularly because these repurposing studies already have, at least in my view, a bit of a bad reputation. Pre-registering these studies to make sure that this isn't considered just a fishing expedition is really, really important. So what were some of the specific challenges for the DREAM study? It's primarily related to the fact that Alzheimer's disease is really just extremely difficult to study as an outcome in secondary data. It is known to be underdiagnosed in clinical practice. The time of onset is very ill-defined, so therefore always to some extent misclassified. There's a 10 to 15 year preclinical period. The mechanisms are, again, very much not clear and are under discussion. And reverse causation is, again, particularly problematic, as prodromal symptoms prior to the diagnosis might lead to treatment initiation. So our approach, this was already mentioned, was to use two large population-based data sources, the Medicare fee-for-service data from the US and CPRD Gold from the UK. And to the extent possible, which is not always the case given that one of these Medicare is a claims data set and CPRD is an EHR database, we used identical specifications, or at least tried to, given the differences in the data sets, being as close in specifications as possible. We decided, after long discussions, to have 4 alternative analyses with equal priority, and you'll see in a second what they are and what the logic behind this is, to have not a primary analysis, but this set of analyses that are considered to be equal in priority and will be interpreted together. And the analytic choices have been pre-specified and registered on clinicaltrials.gov. This is too small for you to read. I will not go through all the repurposing candidates, but what's really critical, as you know from any pharmacoepi study that you have done, is the comparator choice is really key. Finding an adequate comparator is just so important to minimize confounding. So that means, but also means that not all targets have suitable comparators necessarily available, and not all comparators are available in both of these databases that we used. The basic approach is just a standard approach with a period requirement for continuous data, a washout window for the drugs of interest, an exclusion assessment window for Alzheimer's disease or clear precursors of Alzheimer's, inclusion criteria if relevant, and a covariate assessment window. Before I get into the challenges, I will start with an overview of kind of the basic approach, analysis approach 1 here, that is just a cohort approach that is as treated, follows people forward, and has at the end a 3-month grace period to allow for delayed onset of the disease, because obviously Alzheimer's isn't an acute onset disease, and obviously we could discuss forever about what the right grace period here is, and this, I think, one of the limitations of our study, that we had limited data in terms of total follow-up time to work with, and I think we can get into that in the discussion. So here, analysis 1 gives on-treatment estimates, protects against, to some extent, against exposure misclassification. So to the challenges that we faced, one is reverse causation. We have unrecognized or unrecorded dementia onset prior to the treatment initiation, so therefore the symptoms of this undiagnosed disease may predetermine or increase the likelihood of initiation of certain drugs. That's particularly important for drugs that treat behavioral symptoms that are relevant to Alzheimer's, such as benzodiazepines or antipsychotics, but in principle it could apply to all kinds of different drugs. The second challenge is what I've already mentioned, an outcome misclassification. The time that a diagnosis appears in the claims or the EHR typically doesn't represent the onset of the disease, but is delayed by the time needed for diagnosis, seeking care, and so on. The next challenge is informative censoring, where undiagnosed onset of Alzheimer's towards the end of the study, or at any point in the study really, will potentially result in reduced adherence and therefore missing of diagnosed Alzheimer's that happens after the drug was discontinued, so basically a type of informative censoring. And the last challenge is outcome misclassification. It is the positive predictive values for Alzheimer's are quite problematic. We could have a whole symposium, and maybe we should do this sometime, about validation studies and how to do them in Alzheimer's disease, so I will not go in any details, but it's safe to say that the positive predictive values from validation studies are suboptimal, and that all of these validation studies also come with some challenges, so these aren't perfect studies. I think we felt that the outcome was somewhat problematic, but not so problematic that we couldn't see the study through. So how did we address these challenges? So in analysis 2, we introduced a 6 months lag time to address this issue of reverse causation, where we have unidentified dementia that would lead to initiation of the drugs prior to the cohort entry date. And we also implemented an intent to treat analysis, follow-up of maximum 3 years, to address the issue of informative censoring, which obviously wouldn't apply in the intent to treat setting. For analysis 3, we tackled the issue of misclassification of the outcome onset, where we basically, and again we could debate this time period, where we moved the date of the dementia diagnosis 6 months earlier in the analysis. So if something was diagnosed at the end of December of a year, we moved it to the end of June to account for the fact that the analysis is likely delayed, the onset is misclassified. And lastly, we had another analysis that dealt with the issue of outcome misclassification and suboptimal positive predictive values by combining diagnostic codes with prescriptions for Alzheimer's medications, because they are fairly specific or quite specific to Alzheimer's. Here's just an overview so that you can see those 4 designs together. Obviously, confounding is a traditional problem, and I will not go through this in detail. We accounted for demographics, risk factors for dementia, indication specific risk factors, markers of healthy behavior, frailty and healthcare use, comorbid condition, and co-medications. They're listed here, but I won't go through them in detail. And again, there were, for each indication, there was one large set of variables that were identical for each analysis, and then indication-specific variables that were included for each drug comparator pair. I also touch on this here only very briefly. A fairly standard approach for the analysis, a 1-to-1 propensity score matched cohort with incidence rates for dementia calculated with Poisson-based 95% confidence intervals. Cumulative incidence functions used to estimate cumulative incidence of dementia over time, accounting for competing risk of death, which is obviously a big issue in this population, this age group. And then had the primary analysis as Cox proportional hazards regression models to calculate the cost-specific hazards ratio averaged over the entire follow-up time, with subgroup analyses for males and females, presence of baseline cardiovascular disease, and age under 75 versus 75 and above. And with that, I will hand off to Rishi, who will present some results, showing some pictures from my homes at Rutgers.
Okay, hello again. Thank you, Toby and Jody, for nicely setting this up for me to now go into some of the results that we found. So before moving there, though, I should recognize the DREAM was funded by the National Institute on Aging, and I have some other studies that are funded by industry that has nothing to do with this. So over the next maybe 30 minutes or so, my job is to cover these two papers. Both of them are published. So on the left, what you have here is the study where we did find an association. And on the right, what we have is a study where we did not find an association. And I'll make a case for why both are actually equally important. Perhaps the right one is actually more important than the left one here. And so I should note a couple of things before moving on to the studies. So so we did many other comparisons, as Toby showed the list, and I just picked these two as examples. So there were many studies or many comparisons that did not show an association, so you should not kind of assume that everything that we tried worked out. So that's the first thing. The second thing is that, as Toby was describing, we were aiming to do these studies in Medicare claims as well as CPRD, just as a replication exercise. The challenge there is that some of the analyses were actually not approved by the ISAC, the CPRD ISAC, because of the sample size consideration. So both of these studies actually are only conducted in Medicare. On the left, the rheumatoid arthritis study, CPRD just did not have enough sample size because it's a specialty condition. And then for the PD5 inhibitors also was only restricted to Medicare, again, because of sample size consideration. So keep that in mind. We have other comparisons that used both of these data sets, and those papers are in the pipeline. So just keep that in mind. All right, so let's move on to the first case study of hydroxychloroquine. Again, want to highlight a point that Toby has already touched on, is that many repurposing studies are conducted without any kind of pre-specification or pre-registration and selectively reported. And we, of course, wanted to avoid that. And so all the comparisons that we made were pre-registered on clinicaltrials.gov. You can go on the website and download a detailed protocol for all of our study questions, which were registered in advance. So it's just calling it out that is good practice for transparency and replication as well. So in terms of the study design, Toby described the basic design here. I just wanted to point out that for every comparisons, you of course have to make some study specific adjustments. And so this is for the design for the hydroxychloroquine study. And so here we picked methotrexate as the active comparison here. So we are comparing the incidence of Alzheimer's disease and related dementia between patients who were treated with hydroxychloroquine and equivalent comparator methotrexate. So that's the first point there. That's at day 0. These are all new users, so we exclude anybody who had any prevalent use of these two drugs. In the baseline period, we required them to have no existing dementia because we were targeting incidence of dementia as an outcome. So any indication with a very specific definition, we excluded patients with existing dementia. We also required them to have a diagnosis for rheumatoid arthritis. As you might imagine, methotrexate and hydroxychloroquine both are used for multiple autoimmune conditions. And so we wanted to homogenize the population as much as we can. And so we required them to have a diagnosis of rheumatoid arthritis to ensure that the treatment use was for rheumatoid arthritis, and both groups of patients had the same underlying condition. And then finally, we had the covariate assessment window as in typical pharmacoepi studies that preceded the exposure. So we wanted to avoid explaining away the difference by adjusting for things that may lie on the causal pathway. And so the follow-up window started at day 0, and through the 4 alternative analysis schemes that Toby was just describing, we varied them slightly depending on the challenge that we wanted to address. And so I will not go over this because Toby has already covered this in detail, but again want to reiterate that we thought that there is just so much uncertainty in all these analyses, we could not pick one as the most likely or our primary analysis. And so we said we are going to implement all these analyses as equal priority, and only if we see consistency across these 4 analyses, we would conclude something out of these studies. And so it was a specific choice that we made just because we considered that it's not an easy task to study ADRD incidence in claims data or EMRs. So that's something I wanted to point out, but won't go into any of the details of any of the analysis schemes because Toby had just covered that. So moving on to some of the results here. So we adjusted in this comparison for about 75 confounders. These are just selected ones which are most important. So we had some standard demographics, that's the age, gender, as well as race in Medicare data. We included all the dementia risk factors in all our analysis, so that's what you see here. Any evidence of any association with dementia, and in the literature we included them in all analysis because you want to prioritize inclusion of outcome risk factors in your adjustment set. And for the, we are also very concerned about the confounding by healthy user effects or frailty. So we try to account for that using these markers for healthy behavior. And so this included things like smoking or adherence to preventative screening services like mammography, for example, things like that. And finally, for each analysis, we had a tailored set that we adjusted for. So for rheumatoid arthritis, we adjusted for visit to rheumatologist, for example, a number of CRP tests and things like that to get at the extent of the underlying indication to avoid confounding by indication. So on the left, what you have is the crude table. On the right, you have the propensity score match table. And I would like to highlight really one of the strengths here is that you see the comparison group is actually quite equivalent even before matching. So you see standardized differences that are, for the most part, less than 10 or close to 0. And so this, again, one of the strengths we think is that we are able to pick comparators that are equal. So if you have not a lot going on in the measured confounders, you would hope that there's probably not a lot going on in the unmeasured confounders as well. But of course, after propensity score matching, when you have 50,000 people in each group, you probably push everything to close to 0, and so that's what you see on the right side. So overall, for this comparison, we felt quite good about this going in because we felt like rheumatoid arthritis patients were being treated exchangeably between these 2 treatment groups, as seen in this crude table on the left. I would also like to kind of present the incidence rates and highlight a couple of things on this slide. So just because how common rheumatoid arthritis is in the older age, and how large the Medicare data set is, we started off with a robust population of 54,000 patients in each treatment group, which translated into the number of outcomes that were in hundreds. And so for the first analysis scheme, for example, you have 1,000 cases there in the methotrexate group and about 800 cases there in the reference group. And so you will also notice that so each panel is for a different analysis scheme that Toby had just described. So you'll also notice that there are some differences in numbers, and these this is by design. So we start off with 54,000 analysis 1, so which is just the as-treated analysis, the bread and butter of pharmacoepi designs. But then when we include the lag period, and when we require patients to be treated for at least 6 months to have only patients in our study who were using these drugs for some time, you of course lose some patients. So that comparison only includes 30,000 patients each, because patients who are non-adherent in this first 6 months dropped out of the study. So that is one thing I will note. And then finally, the fourth analysis, which had a really specific outcome definition. So you had to have a diagnosis code along with a symptomatic treatment for Alzheimer's disease. And there you see the numbers drop off, because of course not everybody who is diagnosed with a mild cognitive impairment or Alzheimer's gets symptomatic treatment. And so there you see the incidence rates are quite different from the rest of the 3 analyses. That is again by design. In this, the analysis 4, what we are hoping to achieve is that if the misclassification is not differential, we would still see risk ratios or hazard ratios that are unbiased. And so again, just pointing out that we did not know which analysis is the best, and so we implemented all the 4 with equal priority. So moving on, what we found. So these are the cumulative incidence plots accounting for the competing risk of mortality using cumulative incidence functions, and each panel is for each analysis that we implemented. So as you can see here on if you go from analysis 1, 2, 3, and 4, you kind of see a consistent pattern. And what we see is that the curves stay basically on top of each other for first 2.5 years, and then they start separating out for all the 4 analyses actually. So quite consistent pattern that we see. And for an outcome like dementia, we think that this is really reassuring to see no separation of the curve right away, because if that happens, that probably means that that's not the drug, that's the people, and so you would not believe those results as much. But here we saw a trend that as pharmacoepidemiologists we kind of tend to trust more, because you see some time before the drug shows its effect. And so we see that in all 4 analyses, the hydroxychloroquine curve stays below the methotrexate curve after the first 2.5 years. And this also translates into hazard ratios that are protective for hydroxychloroquine. So on left you have crude, on the right you have propensity score matched analysis for 1, 2, 3, and 4. And as you can see here, we found a consistent effect across the 4 analyses in the range of maybe 10 to 15%. So if you go from analysis 1 to 4, you see confidence intervals are largely overlapping. So very, very consistent, robust finding across these 4 analyses. So in the Medicare data set, this is was really reassuring to us that we saw consistent finding in these 4 analyses. All of them have some strengths and some limitations, but at least they were not kind of systematically showing us different things. It was very reassuring. However, we did not stop there. We took the signal and we actually also went to some ex vivo and in vitro studies to look at what is the impact of treatment hydroxychloroquine is in mice or in cells that are derived from mice. So this is a study, electrophysiological study in brain slices from mice. So what you have here is synaptic plasticity is a marker for compromised brain function in Alzheimer's disease that happens with accumulation of amyloid beta. And so what you have here is that in A is just an experimental paradigm where you have the brain slice and where the electrodes are placed, so that's S1 and S2. On B, that's for the wild type control, so these are electrophysiology signal when you don't have the disease. So when the mice did not have the disease, you have really nice long-term potentiation. So long-term potentiation basically is with electrode stimulation, you have this brain activity right away, and then it stays on for a few minutes. And so when you do have Alzheimer's, this is what happens in C, where you have this APP-PS1 mice, which is a widely used a mouse model for Alzheimer's disease. You don't see the long-term potentiation. What happens is that when you stimulate the brain, you do have an early potentiation, but that dies down quite quickly. And so this is compromised synaptic activity in the brain. But what we were able to show with the hydroxychloroquine treatment is some rescue of this phenomenon of synaptic plasticity. And so what happens is that, of course, this is not going to be as good as the wild type control, because all these mice have Alzheimer's disease, so there is some compromised brain function already. But with hydroxychloroquine treatment, you see that the dark blue circles are regaining some long-term potentiation. So again, another piece of evidence which would be adding to the overall story here is that electrophysiologically, it does make sense to have the signal that we see. We also had a bunch of in vitro studies that we've performed. So this is from various molecular phenotype that are relevant for Alzheimer's disease. So you may have heard of neuroinflammation, which is quite common for Alzheimer's disease. So these bar graphs are just showing that you do see reduced neuroinflammation in this first panel here in microglial cells if you treat them with hydroxychloroquine, which is the orange bars. Same thing for amyloid beta clearance in microglial cells as well. And finally, you have mutant tau cell lines where you have tau phosphorylation, which is also decreased with hydroxychloroquine treatment. And finally, we also went to the next step of providing some mechanistic evidence. So far what we have seen is that hydroxychloroquine seems to work in the Medicare claims data, also seems to work in these ex vivo experiments as well as in vitro experiments. We don't know why it works. And this is our attempt at trying to get at that. So as Jody mentioned in the first section of this, is that JAK-STAT pathway is the presumed mechanism for this drug. And so in this in vitro experiments, in all the three types of brain cells, you have astrocytes, you have microglia, and you have neurons. What you see is that treatment with hydroxychloroquine does reduce the phosphorylated STAT3 enzyme, which is the presumed mechanism. So this, you can see this as an early establishment of mechanistic plausibility of why hydroxychloroquine might have worked. And so overall, I think through these diverging lines of evidence, what we were able to conclude in this specific study is that, of course, from the Medicare data, which we thought were quite convincing from a pharmacoepi standpoint, that hydroxychloroquine treated patients had a lower risk. And then we also brought in these pathogenic mechanisms in AD, including the synaptic dysfunction, which I was describing, neuroinflammation, amyloid beta clearance, tau phosphorylation, all of which provided converging evidence. And so overall, for this study, the paper was published a few months ago, where we conclude that it seems like converging lines of evidence are suggestive that hydroxychloroquine may be beneficial in ADRD, and it should be seriously considered as a candidate for trying out in clinical trials. So this is the first case study that I wanted to get to, where we do have, or we think that we see some convincing evidence of a treatment effect that might actually be there. The second story I wanted to tell is actually a more fun story, is the PDE5 inhibitor. So even if you don't recognize right away what PDE5 inhibitors are, you probably have heard of Viagra and Cialis. So Viagra and Cialis are the drugs that are used for erectile dysfunction, and they act on the PDE5 pathway. And so these are the drugs that were proposed. We were not the only people working on this, but this was proposed as a potential treatment for Alzheimer's disease as a candidate. So I would like to start with a study. Actually, this is not from the DREAM study. So this study was published a few months ago in parallel out of Cleveland Clinic. So we were working on the DREAM study on PDE5 comparison simultaneously, but these people beat us to publication. So this was a study conducted using market scan claims data. They were looking at the same drugs, Viagra and Cialis, and they reported, the text is probably too small, but they reported a hazard ratio of 0.31 for Viagra and reduction in ADRD. And the week this paper was published, this was everywhere. So if you look at the altmetric score, more than 2,600, even this guy was excited with these results. And for my non-US friends who don't know who this is, don't worry about it. But this is Tucker Carlson, of course. He's describing this as a breakthrough in the prime time, in 8 p.m. prime time slot. And this was covered widely, and it's actually a really funny clip if you want to go and Google it. They basically take the findings at face value. They say Viagra is a potential cure for Alzheimer's. So, of course, we got perked up when we saw the study and we saw all the coverage that it had received, and we wanted to take a closer look and see how it was conducted and what might explain this really phenomenal finding. And so in the Fang et al. Study, we went in and took a deeper dive, and in the next 2 to 3 slides, I am going to present what we saw there, which is very interesting. So market scan claims were used. So market scan claims, for non-US friends, it's very similar in structure to Medicare. It's just capturing a different type of patient population. So these are working adults who receive their insurance through their employers. And so you do have some patients who are older than 65 who are still working or whose employer is still covering for the healthcare, which is different from Medicare, which is the federal program. So in the Fang et al. Study, they looked at Viagra or sildenafil as the exposure of interest, and they conducted 3 different comparisons. So the first comparison was with a non-user. The second comparison was with 2 anti-diabetics. And the third comparison was with 2 antihypertensives. And in terms of their design, what they did, they conducted a new user cohort study, which is on the right. This is from their appendix. A new user cohort study. So this is the Viagra users. This is the control. And they started following patients up after initiation, which is good pharmacoepi practice. And then they continued follow-up until discontinuation. The confounding adjustment, let's just say, was not robust. They only accounted for 6 factors. It's a bare minimum set. They included the 6 factors in the propensity score model, and then they matched on the propensity scores. And then they also used the Cox proportional hazard regression model for the outcome association. And these are the results, not the Tucker Carlson version, but the paper version. It's super interesting to look at this for a couple of reasons. So you see the hazard ratios for all the comparisons. They're kind of hugging the 0, right? So you have consistent evidence, of course, for all the comparisons. So this is the non-users. These are the 2 antihypertensives, and these are the 2 anti-diabetics. You have hazard ratios that range from 0.31 to 0.45, so solid protective effect. And this is where the 69% reduction is coming from in the news articles. On the right, what you have are the Kaplan-Meier plots. And as you see, for all the 5 comparisons, the effect is almost instantaneous. So after a few months of taking Viagra, your Alzheimer's goes away or doesn't happen. So these are their 5 Kaplan-Meier plots, and as reported in the journal article. The p-values you see, of course, are minuscule, which you can also estimate from the hazard ratio and confidence intervals. And so, of course, as pharmacoepidemiologists, we know better, right? We know that these are problematic findings. So what is problematic about them? So the first issue is that you have you have an active comparator study, but if your active comparators are not treating the same underlying indications, it's not doing anything for you. Actually, they may have made the situation worse here because they are comparing an erectile dysfunction drug to an anti-diabetic drug, and diabetes is a well-known risk factor for Alzheimer's disease. So they are just enriching their control population with people who have higher risk for Alzheimer's to begin with. So that is the first issue. The second issue is that most people taking Viagra for erectile dysfunction in the older age, it probably is an indication that they have some physiological reserves that are preserved. It's a marker for some healthy some health in these patients. So it's really a fertile ground for healthy user bias, because you have patients who are 60 years of age going into a physician asking for a Viagra prescription, and then you're comparing them with non-users, and you don't even know if the non-users have erectile dysfunction or not. So really, really fertile grounds for healthy user bias. And finally, the adjustment set was really bare minimum. They were adjusting for 6 confounders. And so, as we know, in claims-based data or claims-based studies, we frequently rely on this phenomenon of proxy measurement. So even if we think that the underlying concepts that we are measuring as confounders are not directly measuring the confounding, we are hoping that they are measuring something that's meaningful, that is related to the confounders. But if you're just adjusting for 6 factors, you are just really leaving a lot on the table in terms of residual confounding. So anyway, I don't think I need to preach anybody here about issues with studies like this, right? So every time we come across this, it makes us really disappointed. And the fact is that there are articles like this. This is just one example. There are articles like this coming out mainly in science journals like Nature Aging, which is the journal that this was published in, but also in some clinical journals, because everybody likes headlines and everybody likes to get on Tucker, right? So it's a really sad situation there. And what made this extra specially sad is that there are just crumbs in the paper itself, the published paper, that are showing the issues with the study. So this is, again, I did not make this. This is from their published paper. So what they did was subgroup analysis with sex. So you have males and females. And what you see here is that just by that restriction from between males and females, females, of course, are not taking Viagra for erectile dysfunction. They are taking it for the pulmonary arterial hypertension, which is the second indication. You get rid of a lot of the confounding right there. So if you see all the blue results are highly significant, and then just with the 6 confounders that they adjusted for, you move the estimates really close towards the null. And so this is the first indication that the study is problematic. The second indication is that the propensity score overlap plots, of course, when you are comparing a treatment that is used for a completely different indication, the scores are not going to overlap because their characteristics are going to differ quite widely. And so, again, this is published in the actual paper. It's really shocking to see that this made it past peer review. But this happens every single day. So, again, not to bash on the study too much, more than what I already have. How did we go about this in the DREAM study? So after looking at the study carefully, we felt quite confident that this is really a bad study, and what we were doing is much more high quality in terms of trying to ensure the rigor in the analysis. So how did we try to address some of those issues? Well, we did not study patients who were using Viagra for erectile dysfunction, because we think that there is just so much unmeasured confounding going on there that it really makes it not possible to reliably conduct this study in that patient population. So we went for the alternative indication for Viagra, which is the pulmonary arterial hypertension. So we restricted to that indication. So that was our strategy for addressing unmeasured confounder. The nice thing about that is that there are alternate drugs that are used for PAH, which is the ERAs, which we used as active comparators. And so we were able to kind of find a subpopulation where we think that this question can be studied in a more robust way, if not definitive way, compared to the previous investigations. And of course, we adjusted for many more confounders because we are relying on these proxy variables to bail us out really in a situation where you don't have the variables that you want to have in a claims-based setting. So we adjusted for 75 covariates in this study. As a result, of course, our propensity score overlap plots look like this. This is a good indication that the comparator groups are basically used more or less in a similar way, in a similar patient population. It's a pretty good overlap, which is, of course, not the case in the prior study. And then in terms of the results, this is what we see. So we see across all these analyses, especially 1, 2, and 4, the lines are overlapping for the Viagra and the ERA comparator drugs. And in terms of the hazard ratios, this was also appearing really pretty darn close to 1 in terms of hazard ratios. So what we are basically say based on these Medicare claims only analysis is that there likely is nothing going on there. The previous study that found an association is likely fully explained by unmeasured confounding concerns. We also actually had the results from the in vitro studies in this paper as well. I'm just not presenting them here, but none of those findings were showing any effect for Viagra or PDE5 inhibitors. Okay, so that's, I think, brings me to the slide where I would like to kind of make some general conclusions. Of course, these are just 2 of the many studies that we attempted, but I think we have a good sense of what might be going on. So we will continue to see rise in pharmacoepi approaches for repurposing because it's just so easy to access these data. Anybody practically with an institutional affiliation will get their hands on some form of data. And this really is very much up the alley of these basic scientists who are trying to kind of find new targets and do these kinds of studies. So it's a really volatile situation, I would say. Of course, as pharmacoepidemiologists, we are always encouraging of wider use of routinely collected healthcare data. But really, you have to take a step back when you see findings like a hazard ratio of 0.31 and the Kaplan-Meier plots that separate right away to just say that this is really potentially just noise in the data and not something that is biologically plausible. So careful interpretation, I think, in general is a great idea, especially for repurposing studies when you have a lot of potential for overinterpretation here. And joining forces with other disciplines, we've learned, is a really great idea to generate these converging lines of evidence. So I cannot do the electrophysiology study even if I wanted to, right? We are relying on an entire team to support these activities, and really has been an eye-opening experience going from a hypothesis generation, which is completely based on omic studies, to pharmacoepi studies, which is what we do, to some mechanistic validation, which follows the pharmacoepi study in a really logical pipeline of how things should be done. So I think that overall, we are really proud of what we were able to do with the DREAM study. I think this is perhaps one of the examples of rigorously conducted pharmacoepi investigations, which are all pre-specified analysis, pre-registered protocols, and we still have the final paper to write, which should be coming out hopefully at some point in the future. But we have really grown as researchers, I think, trying to address these drug repurposing issues in combination with other fields, other disciplines. So these are my general conclusions. And the last point I wanted to make is the really important one, which is if you just split out the hypothesis generation from hypothesis refinement or validation, where 2 things are occurring at separate places, you really reduce the potential for cognitive bias. Because if I come up with a hypothesis and if I don't confirm it, I'm sad. But if someone else comes up with a hypothesis and I don't confirm it, it's fine. It's just something that we tried and did not work out, which is actually the case for many or most of the comparisons that we attempted. So I think these are some of my general conclusions. I wanted to thank you all for staying late for this presentation, and we will have a lot of interesting, hopefully, questions and discussions. And to stimulate discussion, I actually asked all 3 of us to come up with 1 point, and I am not going to cover these, but just throwing them up there, so hopefully we can address in the question and answer. So Toby, Jody, would you please join us? So all questions are welcome. You share your experience doing repurposing studies if you have done any of them. Yeah, anything. Yes, please.
It was terrific. Great
Thank you.
presentation by all of you. I loved your last comment about coupling hypothesis generation and testing.
Mhm.
When you were planning this, you were handed a list of like 16 drugs
Yes.
to look at. How did you approach that issue of multiple testing of those 16 drugs as a group? How did you get there?
Yeah, that's an excellent question. So we did not. So we tried to keep it exploratory in the sense that we were going in with a biologically plausible analysis. So even if we did find, it could be, of course, false positive, but because we had this biological rationale for pursuing these analyses, we felt like multiple adjustment for comparisons was not needed. Great question, thanks.
Hi. Not to beat up on Fang and colleagues, but
I hope you don't know them.
well, I don't. But
I do know them.
kind of a low-key flex. But we actually wrote a letter critiquing that study, and it took a year to actually get published.
Yeah, UNC, right?
Correct.
I read your letter, loved it.
I hope none of my mentors are here, but sure. I guess like in our letter, in my original iteration of the letter, in their computer simulation, they did not find Cialis as a potential candidate to for Alzheimer's disease. And in our original letter, we proposed Cialis as an active comparator. My question is, in your empiric study, did you guys think about doing that head-to-head comparison? And if your collaborators felt differently about it from a computational biologist perspective?
Yes, so in the omics study, actually, it was not the drug itself, but it was the drug class which was considered to be associated. So that's what, so we treated them as a class. We did not.
So they never thought about comparing Viagra to Cialis at all? That was what I proposed in the letter.
Yeah, we did not do that.
The hydroxychloroquine acts very different, a very different mechanism of action than methotrexate in rheumatoid arthritis. So could it be that it's the way how it ameliorates the symptoms of rheumatoid arthritis is actually improving Alzheimer symptoms for a certain period of time? And I missed somehow in vivo data in animals where you apply the hydroxychloroquine to the food or to inject it, whatever, because I'm not sure how much it crosses blood-brain barrier actually. It is relatively hydrophilic. It acts by the endosomes, that it increases the pH of the endosomes. So I guess it's rather hydrophilic compartments. I'm not sure how much goes in the brain.
So very good questions. I can take the second question, maybe you can take the first question about the mechanisms. So to your question about the hydrophilicity, I think it is true that it does not cross the blood-brain barrier as quickly. So if you notice the dose, we had to go with a 50 micromolar dose, which is actually, if you compare it to human dose, is quite high. And so for this purpose, we thought it was probably okay, but very good point. You want to comment on the first question?
I don't think I really have a response. I'm not sure why the symptoms of rheumatoid arthritis would affect the outcome measure.
I think because it's inflammation. It's believed that inflammation plays a role in Alzheimer, and when you treat inflammation better with hypolysis in a specific way, right? Because it's very different from a mechanistic point, and probably the cytokine profile is different between hydroxyzine and methotrexate, the silencing of the cytokine.
But perhaps on the causal pathway then?
On the causal, yeah, I mean, it's still interesting.
Yeah.
Because of course, only that would mean that inflammatory factors rheumatoid arthritis influence moderate Alzheimer, and that's still very interesting. Is anybody interested?
I don't have anything else.
Go ahead.
Hi, this is Sarah from Merck. I have a question about the rheumatoid arthritis study. So we know that these two drugs, they may have very different dosing regimens. So like methotrexate is given to patients like once per week. But hydroxychloroquine, I think, is given to patient every day. So is it possible that the physicians, they have some preference that they give methotrexate to patients who with like maybe worse cognitive functions, but give hydroxychloroquine to patients with better cognitive functions?
Yes, so it is entirely possible. However, in one of our studies, we tried to get at that by including a 6-month lag and making sure that people are adherent on these treatments for at least 6 months. So that's analysis 2, which was consistent with the other analysis. So it, of course, is possible, but we did not find a convincing evidence otherwise.
So did you measure? I mean, the baseline like patient cognitive functions. I mean,
So that's the challenge in Medicare claims is that you really don't have any good markers for that, and so it's it's a challenge.
Thank you. So, sorry, I have another question, another methodology question. So I see that you use like cohort study to do the drug repurposing study. I think it's very intuitive. I like your comments about maybe doing some case control studies for drug repurposing research. For example, in this case, we have Alzheimer's patients and patients without Alzheimer and go back to see their differences in drug exposure.
I'm sorry if I implied that. That's actually not we think is a good idea, because when you have the outcome that is like cognitive impairment, I think it could have an impact on your treatment adherence. So what you might see is that in the closer time periods of a diagnosis, you may see that people are not using the drug just because the adherence is compromised. So I think case control for these types of studies is not a good idea. And we actually covered this in the first paper that we wrote in 2020.
Hi, Felix from Karolinska at Stockholm. Thank you for the presentation. A question about general in drug repurposing. I'm thinking a danger could be the other indication that the drug is used is not, the treatment is not sufficient to reach the off-target effects. An example could be, what if sildenafil has an effect on Alzheimer's, just not when it's used intermittently, where you have to reach a higher dose and continuous use. And you mentioned you have several other drugs which you looked at where you had negative findings. Have you considered this in those situations?
I think that's absolutely a possibility that we miss. I think the biggest, miss true effects. I think the biggest issue in the context of Alzheimer is the timescales that we're thinking of. So we are looking here at relatively short treatment durations in context to the onset of Alzheimer's. In theory, all these drugs that we use that have plausible mechanisms might have effects if you look at people in their 40s taking these drugs, and then for 10 years, and then you look 20 years after this. There are obviously data challenges with this that certainly in the Medicare data we couldn't do this. And even if you had the data, these studies where you have long timescales become more difficult for all kinds of other reasons. But I think that, to me, is probably the biggest limitation, that we only look at this relatively short timescale in a disease where we have pretty good evidence that the preclinical phase is quite long.
Hi, Steve Schachterly from Pfizer. Obviously, drug indication finding and drug repurposing is of great interest to us. And with all these different exposures, all these different pathways and outcomes, inevitably machine learning will creep in, and there'll be, there is literature on that. I'd be interested in your thoughts on where some of those AI ML approaches fit in the sort of indication, sort of signal detection versus refinement spectrum, and also just sort of how the machine learning world and pharmacoepi can be bridged with, just sort of better implementation of both methods.
Sure, yeah, so I can try and take that. So there is definitely a lot going on with these machine learning type approaches in the omics world, right? So they have just vast quantities of data. It's not the vast quantity of data that we have, it's different. But there is a lot going on there, and I think that's probably the place for machine learning is in that hypothesis generation step. I don't think, I mean, there are enhancements that you can do in the pharmacoepi space with machine learning, but I think that it's really the underlying design and analysis that has to be robust. So we haven't really considered that in terms of the refinement or validation studies, but definitely in the omics space. Actually, Madhav and his team has some studies or some work that's coming out using those types of approaches.
Hello, so I'm Cherry Joey Wu from the University of Toronto, and in your hydroxychloroquine study, and you mentioned that the hazard ratio separate, I think after 1,000 days. So I was wondering if we actually see early separation in this type of study, would you say that it's more likely to due to bias, or would you completely say that, or would you still hold the possibility that the drug still have a potential to be repurposed as at least in the case of dementia?
Yeah, so I can take that as well. I think that any time you see an early separation for an outcome like dementia, I think that gives you a pause. The reason why is that it does take some time, we think, to first the drug to have reach some steady state in your body and produce an effect, and then dementia will not kind of appear right away. And so we feel more confident if we see results like that, where you don't see anything for a year or two, and then you may start seeing a separation.
Sorry, just a follow-up. So, and in your population, I think the mean age is around age 73.
Yep.
Yeah, and then this is actually very close to the preclinical stage of Alzheimer's disease.
Yeah.
And then it's also possible that some drug can interact with the pathology that has been developed, and then it can possibly produce an effect in the early stage of disease. So.
Yeah, no, that's a fair point. I think there's just so much we don't know about these drugs. Right?
Sorry, one more question, and then, yeah, I noticed that in the case, I'm doing research in neurology, and then in the case of neurology, it seems that it's a lot of study with doing the repurposing, and then it seems that the editorial team usually is unable to differentiate right versus or invalid comments. So it's good that you are working on this, so it's advancing the field. But I was wondering how you deal with the editorial comment, and or how you persuade the author to consider a more advanced method to mitigate these different sources of biases.
Yeah, so it's a really pain point. So you really don't have a good solution here. In my experience, I do get invited to review these papers. What happens is that I say this is not good, reject, and then that journal might reject it, but it shows up somewhere else completely unchanged. So I don't think that there is a great solution to this problem. I don't know if Jody has anything to add.
Well, I think the solution is that we all have to say yes, right? When we're sent papers to review, we have to review them, right? We should all be doing 1 or 2 papers a month, right? To be contributing to this community.
Yeah, and it's actually really interesting. It's not like the marginal journal. So Nature Aging is relatively newer and not as kind of prominent as Nature. But a few years ago, Science, the Mother Science, published a study for Parkinson's, which was also highly confounded by smoking. And I think the Danish group actually published a study refuting that in neurology. So it's a big problem. You did, yep. Thanks, Anton. So yeah, it's a really problematic space or world that we live in, because data access is just so easy, anybody could get their hands on it.
And I think one of the other, just to follow up on this issues, is that the original study is on, Tucker Carlson show, which, might not be the best place to do. But the press attention for the original study is just always a magnitude higher than for the refuting study that comes out, 6 months or a year after.
Our,
And I think
our altmetric score is 300 versus 2600.
Thanks for the great presentations. My name is Shahab Abtahi from Utrecht University. I was actually wondering, so can we call these whole methodologies a triangulation of evidence? It's from epidemiological data coming from pharmacoepi studies and also omics data that you showed us. But I was wondering why you started from the omics data. So isn't it easier to start from some signals found in real-world evidence, pharmacoepi studies? And if we talk about not beneficial effect in this case, if we talk about adverse effects, what is the role of pharmacovigilance data? So
Yeah, that's actually a really good question. You want to?
Well, Dr. Thambasadi is a clinical pharmacologist, so that's where he started, right?
Yeah.
But I think that's a good point, right? You can come in from different points to those
Although
paths.
there are philosophical arguments and commentaries that are written about whether you should use the same data for hypothesis generation and testing. I mean, in our case, we could have done Medicare and CPRD separately, but in general, I think if you have a strong biology to go with the actual hypothesis refinement step, that would be my preference, because otherwise you may be picking up signals that may or may not mean anything biologically.
And I think the important thing is to really do the work on talking to my pharmacology colleagues for these in vivo studies, and rather than just relying on a hypothetical mechanism, because I want to say it was an old colleague, I haven't seen him for a few years, Jacques Laurier, who said at an ISPI meeting that a good pharmacologist can give you a biologically plausible mechanism for any exposure and any outcome, and the opposite in a morning. So I think it's important to not stop at just mentioning that there is a plausible mechanism, but go in that next step.
Hi, I'm Jonathan from GSK. I have two quick questions. I mean, first, this is a really great example of taking kind of signal finding and really driving it down into deep epi, of which I don't know that there's a lot of good examples. But I was curious about one thing. The growth in omics data, specifically proteomics data, one of the considerations that is coming up is around when the sample is collected in relation to the disease state, right? And that's a big issue of what's the right time. Is it 5 years before? Do you want it at the time of diagnosis if you can? If it's collected 5 years later, does it matter? I wanted to know if you're aware of the source data when the samples were collected in that main study?
Yeah, so I do have some sense on that. So So BLSA, Baltimore, the parent study which they used for omics, they did actually a case control match where they had patients with AD and then some controls of the same age, and they did try to account for various characteristics to find these signals.
So they were comparing the omics of people basically after they'd already developed the disease.
Exactly.
Okay. And then
Because they received that brain tissue sample of people after they die, right? So there is just not much you can, right, like when they are alive, there is not much you can.
Well, it'll be interested to see what this looks like in some of maybe the more randomly selected samples, from UK Biobank coming out, et cetera, and see if you start to see similar trends. The second question I had was around ApoE. So I don't think it would impact any of your findings for methotrexate versus not, because if ApoE risk is associated with RA risk, it doesn't matter because you're comparing within RA. But I do wonder if there's a risk for an interaction, potentially, and whether you've explored whether there's any evidence to suggest that maybe it's ApoE and methotrexate that might have some interaction.
I don't know the answer, and we certainly don't have, we don't have that in the claims data, so that would be hard.
Right, okay, cool.
Good point, thanks.
Thank you.
I think I have a question coming more to sort of the philosophical approach to things on your final point in terms of the being independent from the original conception of the idea, and that you are more free in that aspect. You mentioned the Parkinson study by Scherzer and colleagues in Science. We decided to invite those authors to our replication study, where we really replicated their bias and interpreted this very differently. And as you know, they are not co-authors of the final publication, and that did not go down in any pretty way when we interpreted it differently. But could you perhaps comment a bit on that aspect? Because I think you had some very nice thoughts of this sort of arm's length from the original conception. How has that worked in terms of having such a collaboration with basic scientists, some more invested in some of these hypotheses? I think this is a really important aspect.
Yeah, so I can comment on that. So this is actually my second rodeo. So the first study that we did was not actually specific to Alzheimer's. It was published in Nature Communication with Sebastian many years ago. And there we actually really benefited from being in a community at Harvard, which is really a hub for various types of activities. So we did not come up with that actually. We were approached by basic pharmacologists saying that you guys are doing such cool stuff with data, patient level data, and we have a bunch of signals for you to look at. Why don't you go ahead and try that out? So that's how that began. This was very much the same with the Alzheimer's disease as well. We were approached by clinical pharmacologists. So I think it's just raising, and I think it also helped that Jody actually had ongoing discussions and connections with Madhav to just let them know that there really is a community out there that can help them, really. And so I don't think there is a solution except for raising awareness of us as a field and as a really important players on the table.
Then what about Madhav's role throughout the process? I mean, he was involved, right?
He was involved, but we completely conducted analysis separately from them. So they did not have any say in whether to go ahead with the publication, or they did comment obviously on the design and stuff like that, but we hope that that's not kind of introducing any biases, because we had the ultimate say actually as pharmacoepi people.
And I mean, I think we had kind of spirited discussions with Madhav when it came to interpreting the results. Again, you saw these 4 analyses, and there clearly was the case that he would look at, an example where analysis 3 was borderline, protective and the others weren't, where he said, but isn't there something there yet? And, it took, mutual respect and taking the time to really walk, him and his colleagues through, why we can't just run with this and say, analysis 3 is positive, so why not publish that? So I think when entering these collaborations, it needs to be clear that there's a mutual respect and not a, financial or otherwise reliance where one person has the power to make the decision. So, and fortunately in this situation, I think we had a really good relationship, but it took some time to kind of cross-educate. I think we spent, fortunately, more time educating him, because it would take me too long to understand what Madhav is doing, but it was a really rewarding process to learn about their work and to educate.
Yeah, and actually,
vice versa.
There's one more comment I will make. So the list that you see in this paper that was published 2020, the 15 drugs, that was not actually the entire list that we received. So we received 50 or maybe 100 drugs or something like that, and then we screened the majority of them out, saying that it's not feasible, either because it's just not enough in use, or the underlying indication is just such a threat to confounding, is that we are not able to do this in a reliable way in claims data. So it was a lot of back and forth and education on, but it's an interesting exercise.
Hello, thank you for the presentation. I'm Laure from the Bordeaux Pharmacoepi team. And I was just wondering, with regard to the misclassification problem for the outcomes, if you have considered using cohort data linked to medico-administrative data, where you would have a good definition of the outcome and probably a long-term follow-up for cognitive decline, and finally the dementia or Alzheimer's disease?
Yeah, those data are hard to come by, unfortunately. It's just
big enough.
It's not big enough. So we are able to link to our hospitals data, for example, in Boston, 2 hospitals. I don't think you can do any meaningful pharmacoepi especially when you are talking about signal that are just not as strong and conditions like rheumatoid arthritis, which is relatively not as prevalent.
Yeah, you think you don't have enough power? You mean not enough subject? Okay. Oh, thank you.
Leo Russo from Pfizer. So great presentation, and you got me really thinking on how confounded by indication, selective prescribing is key. What's your recommendation or view on off-label use of a drug? So if you've got a drug of interest, and it's you clearly see that it's being used not for any of its indicated or on-label uses, but in the non-label uses. I'm thinking there's got to be some biases there. Do you, as a rule, usually exclude that, or do you sometimes, is it a case-by-case basis and you consider it?
I think for most of these we required a diagnosis.
Yeah, so we excluded the off-label use, or actually we excluded or we restricted to just one indication for most of these conditions, because we think that confounding adjustment by restriction is a really strong way to avoid confounding by indication.
Okay.
Yeah, so we avoided that for the most part.
Thank you.
Perfect.
Ken from UNC. A quick question on the data sets. So for CPRD data, I don't have much experience, but I can imagine maybe more clinical details, a longer follow-up, or better outcome definition when assessing drug effects for dementia outcome. And could you talk a little bit more about this? What specific advantage can this CPRD data bring to the table?
Toby?
Yeah, so there are two things. So we used for CPRD specific outcome definitions, which have some validation studies that give you better positive predictive values. In terms of really, in terms of other adjustments, we were kind of struck between trying to keep the approach close to the Medicare analysis and taking advantage of the additional information in CPRD. Practically speaking, really, we had only kind of a handful of analyses where CPRD had sufficient sample sizes, unfortunately. And obviously, we had to make some adjustments. For example, in the Medicare, we have a one-year look back for comorbid diagnosis for the assessment. In CPRD, we expanded this to any time before for chronic conditions, because in the electronic health record, diagnoses aren't just repeated at any visit. So kind of those types of adjustments we made. But we didn't, in some ways, we didn't do the CPRD study as good as we possibly could have done, because we wanted to maintain the comparability with the Medicare data.
All right, well, we are almost at time. If there are no questions, let me just thank again all of you for staying until the end, and our panelists for really excellent presentations, and concluding the DREAM study.